Now we will spend some time understanding a second kind of cell division, which is called as meiosis. Meiosis occurs only at a certain times in the life cycle of all sexually reproducing organisms, whether they're plants or animals. In humans, meiosis occurs only in the reproductive organs, and it results in the formation of gametes. Gametes are what is called as the sex cells. So let me get this out. We're a little off on the stage. Okay, gametes are are the sex cells, and before we go on to understanding meiosis, I want you to ponder upon this concept. If the sex cells, for example, in male and female for humans, have a certain number of chromosome, and humans have 46 chromosome number. So the fertilized egg by the sperm is called as a zygote. The zygote is the first cell that is formed with the union of a male sperm and a female egg. Now, if the zygote was to fuse these two chromosomal numbers, then the zygote would have a chromosome number of 92, and that will not maintain the species of human beings, because it's way it's almost double the number of chromosome. So in order to avoid this confusion about how to maintain the chromosomal number, this kind of cell division takes place. And actually, meo means less, so it's a kind of cell division in which the chromosome numbers are reduced. Keep in mind, meiosis is only taking place in sexually reproducing organisms, and in particular the sexual reproductive organs. Okay. Now let's look at the most important characteristic that takes place within meiosis is the formation of what are called as homologous chromosomes. Homologous, as the name tells you, means similar. They are corresponding, they look alike, they have the same length, they have the same position of the centromere, that is the region that connects the two chromatids. So these are two chromosomes, and each chromosome by itself is formed by either the male maternal side of the cell or the paternal side. So you have these two chromosomes, as you can see over here. Each of these chromosomes that you see are called as homologous, because you can see that each of them, they look like they have the same shape on the top. They have the same length on the bottom over here. This is the central piece, which is the centromere. So these chromosomes are homologous chromosomes, and since we have 46 chromosomes, there are 22 homologous chromosomes. The last one pair is the sex chromosome, and the sex chromosome is going to be either two X's or an XY combination. Other than that, these are the two chromosomes that you will find that the homologous, if it's an XY, it's going to be, one's going to be a little longer, and the Y chromosome is going to be a little shorter. Other than that, the other 22 chromosomes are all they all can be paired up, and they have the same length. Now, this concept of homologous chromosome was not present in mitosis. We did not talk about homologous chromosome. What we said under mitosis was that just before cell division, during the S phase, which was the phase where chromosome was duplicated, and then eventually when the chromosome got thickened, it formed a chromosome, and after duplication, it formed it had two chromatids. If you remember, we said use the words two chromatids, and they were joined by the centromere. Now, as it's shown over here for you in two different colors, blue and red, and the reason is that each of the each of the chromosome that is coming from one side could be from maternal and one side could be paternal, is actually uh, then then pairing up to form this combination, which is called as the homologous pair. Now, a homologous chromosome, if you count the number of chromatids, it actually has 1, 2, 3, and 4. So it's actually 4 chromatids, and sometimes it's also called as the tetrads or bivalent. Bivalent means that each each of the bivalent is bivalent consists of the term that there are 2 chromosomes that are joined together, and they're called as bivalents, or 4 tetrads and 4 chromatids, and calling as tetrads. So there are 2 different terms, and they are referring to the same homologous pair. So either bivalent or tetrad. Tetrads are probably easier for you to see that there are 4 chromatids, and that's why it's called as a tetrad, and bivalent because each valent chromosome is then paired up to give you the bivalent concept. So now, many books will ever actually show you the chromosomal mapping, and in chromosomal mapping, you can see images of the chromosome that look like the ones that are shown in this picture, where you have chromosome number 14 that is paired up, 15 paired up, 16 paired up, 22 paired up, and so forth. Now, the information that is contained within each chromosome is actually the genetic information, and that's where the genes are. And another term that we want to understand over here is the term alleles, which means that it could be the version of the same gene. For example, a person may have black hair, and this gene may be present on this part of the chromosome, and this is giving you black hair. On the other hand, the other locus on the other chromosome may actually have blonde hair. But because black is dominant, we will learn later on in the genetics chapter that black would be the expression of this individual. However, keep in mind alleles are the 2 versions of the gene. Sometimes if you have the same allele, for example, if you have black and black on both chromosome, then it's black is dominant, and it's more like a pure form of the gene because there is no mixed genetic makeup. So this term of homologous chromosome was not present in mitosis. Now, meiosis is called as a chromosomal reduction because keep in mind I said meiosis would only take place under sexual reproductive organs, and it results in the formation of sex cells, which are called as the gametes. The sex cells could be the sperm cells in the males and the egg cells in the female. So again, the sex cells are not continuously produced throughout the life cycle of a human being. So it's only during the time of these formation that meiosis will take place. Meiosis, now that we've understood that it is associated with sexual reproduction, now the next thing we want to make sure is we understand that meiosis is actually composed of 2 parts. There is one part that is called as meiosis 1, and the second part that is called as meiosis 2. Unlike mitosis, where we only had one series of 5 stages that was going on, there was only one mitosis. Meiosis is part 1 and part 2. At the end of mitosis, if you recall, a cell divided to give you 2 daughter cells. However, at the end of meiosis, we get 4 daughter cells. So there are 4 cells that will be formed at the end. Now, another very important characteristic that we will learn later, the 4 cells that will form are not identical. Again, this is unlike mitosis. In mitosis, the 2 chromatids separated, and we had identical chromosome in the new cell. But this is going to be not identical, and we will see more of this on the next coming slide. Another overview feature for meiosis is the 4 daughter cells that are produced are haploid. If you recall, I gave you the term haploid and diploid. So originally the cell is 2n, which is twice the number of chromosomes. For example, when we said 46, that would be a 2n or a diploid cell. Now this diploid cell will undergo meiosis and result in the 1n, or the other term for that is the haploid. Haploid means half the number. So these are some of the comparison from mitosis to meiosis. Now notice this is an overview of meiosis. So I'm going to give you a quick one over here, and then we will look at each stage at which a little bit more depth. Now the names of the stages are very similar to what we learned earlier. We will again hear the term prophase, prometaphase, metaphase, anaphase, and telophase. But it's important that while you're looking at the stages, you should now look at the chromosome and its alignment. So the first thing, if you notice in this first image, which is the diploid chromosome, you can see that for simplicity, it's they're shown in two different colors, blue and red. So you can see the two long ones and the two short ones. Now the two long ones are going to they have duplicated, and after duplicated, each of the bivalent is forming a homologous pair, or as it's called, as a tetrad. So you've got one over here, and then you have another one over here. So you've got this unique combination of homologous chromosome, which was unlike mitosis. Now this is the part that is still called as meiosis 1. Now at the end of meiosis 1, this diploid chromosome is going to separate out each of the homologous pairs. So notice now we have a haploid cell which has half the number of chromosomes. So we have now eventually after this meiosis undergoes a second part which is called as meiosis 2, and meiosis 2 is actually a replication of the previous cycle that we learned, which is called as mitosis. In the end of meiosis 2, the two chromatids are now separating and resulting in 4 haploid cells. So notice that you've got one chromosome in each. So if you began with 4, at the end you only have 2 on each cell. So this is a quick overview of the entire cycle. We will look at the cycle in much more depth in the next coming slide. So let's go on to the next slide and look at each stage a little bit more in depth. So the firstly is, why are the daughter cells not identical? This is a major difference in meiosis. In meiosis, what happens is, if you notice over here, the two sister chromatids that are shown are aligning up. Now when they align up, they align each of the bivalent is aligned to give you 4 tetrads. Now at this time, a very unique combination takes place, which is called as crossing over. Just like the name tells you, it is actually a stage for the cell in which parts of the chromatids are actually swapped. That's actually the right word. It's just segments are broken and they are swapped from each cell to the other. Now this results in different combinations at the end. So the process of crossing over takes place in 3 different steps. The first part is called as synapses. Synapses mean it's joining together or forming connections. The synapses takes place within the bivalent chromosome. And the very important thing is the phase that this takes place is called as prophase 1. So if you recall what was happening in prophase, prophase was usually identified with the stage in which the centrioles were moving towards the poles, the nuclear membrane separating, the nucleolus disappearing. So within this part, when the homologous chromosomes start forming these crossing over, it shares segments of one part to the other. And eventually we will notice at the end of the cycle, since we will see later on at the end of the meiosis, when the 4 cells are separated, they will be separated with independent assortment, which means that you will not have a cell that has chromosome 1 and 2 together and 3 and then another chromosome 4 and then one cell empty. No, each cell will separate out independently, and you will have four cells at the end with 4 different combinations. So going back, I'm going to go back over here quickly just to give you an overview of what I'm talking about. Let's see. here we're talking about the four different haploid cells that are coming out because of independent assortment. And in this region, we will talk about the crossing over, or the region where synapses are formed, and so forth. Now, you can pretty much see the significance of sexual reproduction is to give variation in the population. If there was no meiosis, they would be identical clones from the parental generation to offspring, and that's kind of scary thought to think about, that all my kids are going to look like me, and then they'll eventually will be carrying every gene, every characteristic that was from my parents, whether they're good or bad. It's kind of a scary thought, having all your siblings, they look alike, and so forth. So sexual reproduction will give you variation, and as you can see, the variation will result because, as we saw in the previous section, when crossing over took place, segments of the chromosome were shared or swapped, and that will give you genetic variation. Sometimes disease resistance arises due to due to crossing over, and eventually evolutionary process is it results due to crossing over. Now, why create gametes? As I said earlier, the gametes have to have the haploid number, because the gametes must maintain the diploid number of the organism or the species. And if there was no meiosis, the diploid number would double every generation onward. So you'd have 46 unite to form 92, and the next generation would have 184, and so forth. So there would be a constant increase in the chromosomal number. And as in biology, we believe that each species is maintained because of its chromosomal number. So, for example, these cute little puppies that are formed here, they will have the same chromosomal number as the parental as the parent dogs that are up here. So after fertilization, when a haploid gamete from, let's say, if this was a male cell, and this was a female cell, and this was the male cell, and it will give a diploid zygote, which is the first cell that is formed after fertilization, and the diploid zygote would have the exact same number of chromosomal specification. And after the zygote is formed, the cell will undergo a constant division, which is mitosis, which will result in the increase in number of the cell, and eventually results in organization and distribution of different tissues and so forth. So again, keep in mind, mitosis takes place throughout the life cycle of a cell, whether it's to repair or whether it's to increase in number. However, meiosis is very restricted and only takes place within the sexual organs.